Thanks a lot, Tanya. Very nice information. Very nice talk. Um, I have a question. So the slide
Sure.
number 25. So I see you use a rhymer in the header of the timetable. The slide
Um,
number 25.
25. Okay, let me
Yes.
go through it. This one,
Yes.
all right.
So
Could your
you
question again?
said that the column number 4, the total number of primers. So it means that you use the BCR amplifier, not the probe you
Correct.
target from.
Yes, for the targeted DNA,
Mhm.
uh, for your information, targeted DNA, we have the V4 chemistry and we have the V3 chemistry. So these both chemistry, we use the uh, amplification method. So we have the specific primer to amplify the gene of interest. But
Mhm.
normally, if you know, I'm not sure whether you got this information or not. Normally, the way Kaizen amplify, we use the single primer extension. Each specific target that we want to detect, you need just only one specific primer because another site we will use the universal primer. So this one allow you to reduce the bias of the primer dimer and also provide you the more specific during the primer design.
Mm-hmm, mm-hmm.
Yeah.
It means that, like for example, you have 633 primer
Mhm.
to
Yes,
amplify,
correct.
and
Correct.
it means that you run the multiplex PCR in one reaction chip only.
Correct.
Oh my God, it's a
Yeah.
lot to
Yes,
optimize
it's
it.
a lot. Or even the comprehensive. As you can see, now
Yeah.
we use the AI to help us to do the calculation and
Mm-hmm.
design for the primer to detect the specific region. Because definitely if you do it manually, it's very, very tedious if you if you have
Exactly.
the gene like
Exactly. Yeah.
yeah, it's impossible, I would say. Yeah, so that's why we have the supercomputer to calculate for the to help us to design the specific primer that can do multiplexing in the same time.
Mm-hmm. So can you give me the name of the software or the paper that provide the open source software to design this primer? Can we have
Um,
this this software?
well, it's the for the for the software, I afraid that it will be confidential, but
Uh-huh.
you can use that one to design the target of the if you have the target of G list and you want to use these tools or this software, it's
Mm-hmm.
available in the Gene Globe. Yeah. Or you
In the
can you
Giro, where is
yeah.
it? What is the name?
Gene Globe is the Kaizen website. If you go
Mm-hmm.
to the, yeah, please allow me to, uh, you still can see my screen, right?
Yes, I can see your screen.
Yeah.
You can
You just type the Gene Globe. Yeah. My internet is slow a bit. Sorry about that. If it's still slow, later on I can send you the link. Okay.
okay.
And yeah, yeah.
Later on you
And
can do.
then you
Okay.
can go to the custom product. So here, if you scroll down, just a moment, yeah.
I cannot see your screen now. I just see the slide at the moment.
Oh, just a moment, just a
It's
moment.
okay. It's okay. Later, you can show me the YouTube now I
Sure.
can see it. Yeah, now I can see it.
Okay. So this is under the Gene Globe, yeah. You can
Mm-hmm.
search Gene Globe, and I believe that it's come to the first link in the Google, and then you go to the custom product here. Okay. You scroll down a bit. You choose the next generation sequencing here. You select the analysis type, whether you want to generate the specific primer for the DNA,
Mm-hmm.
RNA, or you
Mm-hmm.
want to do the multimodal. Multimodal
Mm-hmm.
is mean that you want to detect the DNA and RNA at the same time.
Yeah.
For these three is the specific primer base, okay? But the
Mm-hmm.
microbial here is the hybrid capture base.
Mm-hmm.
Okay, so let's see for the DNA, and then you go through it whether you want to do the custom human, targeted DNA Pro, X-Hype custom microbial, or and much more data. So
Mm-hmm.
let's say that you want to go to the targeted DNA Pro, which allow you to detect the single nucleotide variation, copy number variation, and the indel. Yeah, all the biomarker in the DNA you can do it. Then you click Open Design. Then it will go through the page that allow you to put the name of the gene list. All right. So you just put the name of the gene list there, which could be the um yeah, you have to type something here, and then if you go to the next, I think I will not show you in the next step because it will take time. Then if you click next here, in the next page, it will allow you to put the name of the gene list that you want to design. And also it allow you to pinpoint whether you want to sequence the whole part for the overall exon, the hotspot, or including the UTR as well. It have
Yeah.
that option for you as well. Yeah, so
Okay.
it's quite flexible and comprehensive.
Yeah. So it's free of charge to do the analysis like want to design the
Correct. Yes, for design, in silico design is free of charge. And if you want to save your time and if you don't mind, you can share that gene list to me. I can do it for you because normally I will not use the gene globe, but I will contact to the enterprise team in the US. So you can got the bed file in one day normally.
Okay,
Yeah.
thanks a lot. And then my second question, so in the lab panel about using a long read to evaluate some kind of gene panel, do we have any IVD for the solution, the
No.
IVD solution?
No. Normally
Okay.
the NGS kit, all the NGS kit from Kaizen now is the RUO. We have the reason behind that. As you know that the NGS is developed even day by day these days. All month you will get the new kit emerging from the different company. And let's say that this one normally is reflect by the update of the publication and the guideline from the different type of the data source, including the WHO as well. So to wait until we develop the IVD kit, I don't think it will be on time with the I mean it will be uh, uh, um, immediately used for the patient that really need to diagnose. So normally, I understand that this law is vary country to country. Some country they fix the rule that you have to use the IVD machine and the IVD product to analyze for the oncology until get the precision medicine list to treat the patient. But in Thailand, normally they allow to use the IVD machine, but for the kit, you still can use the RUO. But you need the pre the geneticist to sign, or the doctor who know about the genetics, genetic counselor. I mean, yeah, to sign whether they approve this data from the our software or not. Yeah, because
Okay.
our software normally it just provide you the list based on the very huge data that we available in the Kaizen knowledge base. Anyway, some genetic counselor they also want to screen another time as well before they will choose the precision medicine list to treat with the particular patient. Yeah.
Okay. So we have one question from Dr. Huyền. She asking for the hereditary cancer panel using a long read sequencing. Have you validate on the nanopore system?
Yes.
And then the second question, what is the cost compared with the short read sequencing? Thank
Okay,
you.
interesting. So the first question, have we validated in the nanopore, right? Sorry.
Yes, the nanopore.
Okay, the answer is yes, we validate in the nanopore already. But since this slide, I just got it, I think last 2 days, so I just add it. It's still warm, if you can feel.
Yeah.
So
It's very hot now, very hot. Still
yeah,
hot.
it's very hot. So I I have now just only the PacBio, but the R&D didn't send me the result for the nanopore yet. But definitely, if you can see this slide that I just skipped, sorry about that. Normally the nanopore here, it can run up to the 2 billion base pair. I mean, in the length unit. But it will compatible with the HLA and the hereditary cancer. Yeah. But anyway, you still need to adapt the library once you finish preparing by the Kaizen product. So what you need to modify it is by the enpolishing and a adding here, because you have to do the enpolishing and the A adding at the 3' end for the both side to allow ligate with the index from the nanopore, because normally the index from the nanopore will compose of the protein which will recognize the pore protein is the mobile protein, if I remember correctly. So this one will bring the fragment whatever that we generate the library go through the pore and push the single strand of the pore in through it, so it will fluctuate for the ion that is normally it will go through the pore as well. So with this fluctuate pattern to the pore, it will go to the signal like this, tut, 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 and then the machine will read it for you whether this pattern is gonna be the A, T, C, or G, something like that. So first answer is yes, it's compatible with the nanopore. However, the price, I don't have that information in my hand now. I just have
Mhm.
only for the um just only the number of the maximum sample per run, just that on the nanopore. Yeah.
Mhm.
Mhm.
Okay.
Yeah.
Thanks a lot. Have any question? Mấy bạn có thể hỏi tiếng Việt rồi tụi tôi, nếu mà tôi không dịch được thì tôi nhờ mấy anh chị khác dịch giùm. Đó, đừng có ngại mọi người, có ai hỏi tiếng Việt không? Hỏi tiếng Việt cũng được mọi người. Ở đây mình không phải thi tiếng Anh. Đây mình muốn học cái này chứ không phải mình thi tiếng Anh muốn biết. Đó, bởi vì không cần phải giỏi tiếng Anh đâu mọi người, cần. Đó, tôi muốn tổ chức cái lớp để mọi người học được, chứ không phải vô đây để mình khoe tiếng Anh của mình, không có nhu cầu đó. Có ai hỏi nữa không mọi người? So my last question concerning about the three panel you show about the low risk, intermediate risk, and then high risk of cancer. And I did not see, or I missed anything that about the, I didn't see about the CT CFP9 for liquid biopsy testing. Over here, always using the FFPE. For a low risk, maybe we can use the blood sample, but
Okay.
not the CTCFP9. For
Mhm.
intermediate risk or the high risk, what kind of sample input we using?
It depends on the cancer type. For example, if that cancer is related to the lung, which very difficult to biopsy, you can use the cell-free DNA. Yeah, but
Mm.
definitely the cell-free DNA, you really need the coverage depth very, very high, because especially the early stage, that the apoptosis for the cancer cell is the rate will be very low. So if you want to detect from the blood,
Mhm.
is would have the um, false negative. So we actually have another kit we call the cell-free DNA Ultra. If you don't mind, please allow me to go through that slide for a while.
Yes,
Yeah.
yes, please.
Okay. Okay. Just a moment. Yeah. All right. Here you go. So, first of all, it depends on the cancer type, whether it can biopsy or not. If yes, so it's pretty easy. You can do the FFPE, yeah, which zero
Mhm.
point 0.5 variant of frequency is more than enough. And we have that solution. And the targeted DNA, you can detect we have solution for the cell-free and for the FFPE as well. However, for the early stage of the detection or even the follow-up after you treat with the precision medicine already, you definitely you want to know that the last single cell of the cancer is gone yet, else it will be elapsed or it will be come back. So we have another kit is called the cell-free DNA Ultra. This one some lab even apply for the MRD is the measurable I forgot the name already. Yeah, it's the MRD. Yes. So that MRD we definitely need the correct depth up to the 48,000 and can detect the variant of great frequency down to the 0.1 here. Okay, so we have this kit and it's available for the cell-free. Okay, especially for the in the scenario that um, it's very difficult for the biopsy and you really want to detect in the early stage. But anyway, this ultra depth, you also need the very high throughput of the sequencer. So for the Illumina, you have to use the NextSeq. 1,000 is fine, 2,000 is the best. But 2,000 you have to go with the P3 for flow cell. If you ask me whether MGI high throughput, for example, the MGI 2000, MGI 50, I believe that it also can run, but the number of the sample per run may be down to the just only single number, could be around 6 to 8 or something like that. Yeah.
Yeah, thanks a lot for your information.
No
So, có ai hỏi gì nữa không mấy bạn? Không hỏi thì mình qua phần mới ha, phần cuối cùng, phân tích cái kết quả, được không mọi người ha? Thanks a lot, Thanas. So now we going to the last presentation.
My pleasure anytime. And just want to emphasize again, if you have any question or would like to know about the detail about the kit, and also my colleague just sent me that if you want to know about the price for the long read kit, particularly for the hereditary cancer panel, the local cell team, happy to share with you on that. Okay, so we will get back to you later on that. But for the technical part, please feel free anytime to contact me or my colleague here. I put it in the last slide. Thank you.
Thank you. So now, Siva, on your.
Thank you, Tanat, and thank you, everyone. I think we are on the right track. Just allow me to share the screen.
Yeah.
Okay.